Hello guys, welcome to my YouTube channel. So today we are going to discuss how you can produce some radio level DNA probe by the method of random primer. Okay, so as you can get it from the name, so our objective is to prepare some radioactively labeled probe DNA. Okay, so that means what? A DNA which if you visualize under autoradiography, which should have some radioactivity so that you can visualize it under autoradiography when you do the autoradiography. So the purpose of this preparation is that this probe that you will prepare will be useful for identifying and locating any particular DNA sequence for which against which this probe DNA is made. Okay, that we will discuss it in a separate lecture. But any application of a probe is to find out its target DNA. So for finding out the target DNA of a particular probe, you need to make the probe first, okay, and then you need to label the probe, be it fluorescence labeling or be it radioactive labeling, because when you will let the probe hybridize with its target DNA, you won't be able to figure out if you don't label it. Okay, so for your purpose, so that you can figure out where the probe has exactly it has bind to the DNA. Okay, so where it has hybridized with the target, so that place to make sure, okay, this is the place where the probe has hybridized with the target, you need to label the probe so that you can watch it hybridizing with this target DNA. And this is a method of random primer. Random primer means you have to synthesize some primers with random nucleotides, uh, combinations of random nucleotides, and then you have to hybridize. So we will discuss. Okay, so suppose we start with the preparation of probe. So suppose we take a DNA, so this is our source DNA, okay, and we let it to denature, at the same time. When it is denaturing, we are giving some oligonucleotide primers. So in general, these oligonucleotide primers are all have all possible combination of 6 nucleotides. Okay, so these are generally 6 nucleotides long. These are oligonucleotide, single stranded, and they have all possible combinations like ATGC, GC, ATGC, AG, like all possible combinations only of 6 nucleotides, single stranded, and we are adding it to the source DNA at the same time when the source DNA is allowing to get denatured. Okay, and we are also letting it to hybridize. Now it is a statistical certainty that some of the molecules of this oligonucleotide mixture will hybridize to the unlabeled denatured probe DNA. Okay, so as you can see, these are the places where the hybridization has taken place. That is, the probe DNA has been bound to the target DNA. So these are the places of hybridization. So that means what? When you are randomly synthesizing some oligonucleotides, which are random combinations of 6 nucleotides. Okay, so some of the it will happen that some of the combinations will fall into place, and those combinations of nucleotides will bind to the target DNA. So that is what has been taken. So suppose you have, what to say, 100, you are giving to the mixture 100 oligonucleotides. Out of that, maybe it may happen, because you are giving 100 different oligonucleotides with all possible combinations of 6 nucleotides each. So it may so happen out of the 100, maybe 20 or 30 combinations have hybridized. Okay, so like that. So after this hybridization process has occurred, you give in the reaction mixture, you give Leno fragment. Okay, so this is an enzyme, if you remember, Leno fragment. So it is synthesized from the DNA polymerase 1. It is a specialized kind of DNA polymerase 1, and you give the 4 DNTPs, deoxyribonucleotide triphosphates. Okay, now why you are adding this Leno fragment? Because it will help in DNA synthesis, and it will synthesize DNA with the help of what? 4 DNTPs. Okay, 4 DNTPs, because these are the substrates of DNA synthesis. Now there is something important here. These 4 DNTPs are actually radio labeled DNTPs. Okay, so whatever DNTPs you will be giving here, these should be radio labeled. Okay, so we have 4 DNTPs available. So it can be DATP, right? You can give DATP, you can give DCTP. Okay, so what else can you give? You can give DGTP, and for DNA you can also give DDTP. Okay, but these have to be radio labeled. So I'm giving star above them to signify that these are radio labeled. These are not the ordinary deoxyribonucleotides, okay, triphosphates. So these are radio labeled. So what will happen? So here, as you can see, this is how from the primer the DNA synthesis will take place by the Leno fragment, and while synthesizing it will use the radioactive DNTPs as the substrate. So in general this entire molecule will become radioactively labeled, yes or no? So this becomes radioactively labeled, right? This is radio labeled. Okay, now we are synthesizing probe. Okay, and probe is generally single stranded, so we don't need double stranded this thing. We want to just excise this portion. This portion, if we can cut it and get it, okay, this portion which has the radio labeled portion within it. Okay, the other portion does not have any radio labeled nucleotide, so that is not our lookout. So this portion, if we can somehow extract it, excise it, then our purpose is done. So we will let it to denature. Okay, again denaturation step, and we will get the single stranded radio labeled probe. Okay, so here what we are getting is single stranded radio labeled probe. So by random priming method we can make single stranded radio labeled probe. Okay, and now these probes could be used to find out, okay, whether in another experiment, another set of experiments, so these probes could be used to see if any sequence is present in our target DNA. So if these radio labeled probes go and bind to some portion of the target DNA, that means what? The complementary sequence of this radio labeled probe is present within that target sequence. That is our purpose. Okay, so we will discuss some of the applications of probe and DNA target DNA hybridization in our subsequent lectures.